Good morning, I'm Matt Golden. It's my pleasure today to introduce Sheila Lukehart, who is a longtime collaborator and friend. Sheila originally got her PhD at UCLA and subsequently did postdocs at UCLA and UCSD before coming to the University of Washington in 1979 to work with King. And she has been here ever since, eventually becoming a professor of medicine and associate dean of research from 2003 until 2018. Sheila has really excelled as a researcher and as an educator. She's really a great example of a person with extraordinary focus, it seems to me, that she has studied syphilis essentially her entire career, not essentially her entire career. And she has been extraordinarily successful. As far as I can count looking at her CV, she has been the PI on 8 R01s and many other NIH grants. She has 145 publications focused primarily on the basic science of T. pallidum infection. And really much of what we know about the basic science of T. pallidum, we know because of Sheila. So she helped define the immune response to T. pallidum. She identified the major antigens associated with the bacteria and how the organism varies those antigens to evade the immune response. And she was instrumental in developing the molecular techniques needed to define T. pallidum strains and subspecies. But although she's really a basic researcher, I think one of the things that stands out to me about Sheila is that she's really good at working with clinicians and getting the clinical aspects of of syphilis and the public health issues. She's also an extraordinary educator and mentor. She's had at least 19 pre- and postdoctoral trainees and at least 22 undergrad and medical school trainees. She has been the PI on UW's STD/AIDS Research Training Grant, which we've had since 1978, since 2003. And we would not be able to have that training grant all of these years were it not for Sheila. And I would also point out that as recently as the last week, I personally have benefited from Sheila's mentorship as we've worked together on an R01. So I'm very grateful for all that Sheila has done for me and all she has done for our division and the University of Washington. And it's a pleasure to have her here.
Thanks, Matt. Well, thank you, Matt, very much for that nice introduction. And thank you for the opportunity to talk to you about my favorite topic, which is syphilis. Syphilis is an old disease, as you well know, and can be seen in this woodcut here. It's a disease that in the 19th century and the early 20th century affected about 10 to 20% of the population of the United States and Europe. But today, when people ask me what I work on and I say syphilis, they always say, "Is that still around?" Well, yeah, syphilis is still around, as I'll tell you about, and the organism itself has identified interesting ways to persist in the host, and so that's what we'll be talking about today. These are my disclosures. So today I'll briefly discuss the epidemiology and public health importance of syphilis. I'll give you an overview of the natural history of syphilis, tell you what we've learned about pathogenesis and the immune responses. We'll discuss the mechanisms of persistence, at least one mechanism of persistence, and give you some unpublished early work on vaccine development. So I told you syphilis was still here, and in fact it is on the rise. It's going up very steadily, and these public health posters, which are very recent and from all over the world, show exactly this point. If we look at the United States, on the graph on the left, the y-axis is syphilis cases. The x-axis is 1940 to 2018, and I want you to notice that the y-axis is a logarithmic scale, not arithmetic scale. And there was a big dip in syphilis in the mid-1950s. Well, I should tell you that the orange line is total syphilis, that is syphilis of all stages. And the green line is primary and secondary syphilis, which are the infectious stages. So from a public health point of view, what you really want to look at is primary and secondary syphilis. There was this dip in syphilis, very dramatic, after World War II and after penicillin became widely available. Penicillin's still the treatment of choice for syphilis, and it was very effective in reducing the numbers, and people thought syphilis was a disease that was going to be treated out of existence. But as you can see, that did not happen, and it bounced back and kind of puttered around for 3 decades until about 1990. And let's look at that period from 1990 till now in a little bit more detail. You can see that that big outbreak in 1990 was a heterosexual outbreak. It took place largely in the inner city, largely among African Americans, and some studies in some of those cities suggested that it was related to the crack cocaine epidemic. And when crack use declined, so did syphilis, until the year 2000, when you see the beginning of this massive increase in total syphilis and early syphilis. And you can see from this graph that it is almost exclusively in men. In 2018, 86% of primary and secondary syphilis was in men, and 55% of primary and secondary syphilis was in MSM, men who have sex with men. This is at the national level. And a much lower proportion in women. We'll talk about women a little bit later, but you can see that numbers are going up there. In terms of the rates of syphilis, that is cases per 100,000 population, in various ethnic and racial groups, African Americans continue to have the higher burden of syphilis than other ethnic and racial groups, but the rate is increasing in every single one of those groups. So what's happening in Seattle? King wrote a paper in 1995 about how syphilis had been eliminated from Seattle, but it bounced right back, and so he had to change the title of the paper to Elimination and Reintroduction of Syphilis. But you can see that since 1995 in Seattle, King County, the numbers have gone up steadily. And thanks to Matt and Anna Burskaltz, I've gotten the provisional data for 2019. About 700 cases of early syphilis have been reported in Seattle, King County. And in contrast to the 55% nationally that occurs in MSM, 90% of syphilis in King County is in men who have sex with men. 4% in men who have sex with women only, and 4.5% in women. And if we look at this lower graph, these are the data for women. And you can see there's been a large increase in the past couple years. And every time we see an increase in infectious syphilis in women, we expect to see congenital syphilis. And in fact, in 2019, there were 3 cases of congenital syphilis in the county, 2 women, one of whom delivered twins. And interestingly, both of these women were in prenatal care, had been screened in the first trimester, and were screened negative. So these high-risk women really should be screened in the 2nd and 3rd trimester as well, if they can be identified as high risk, because these women clearly got syphilis during their pregnancy. So I'm going to show you the natural history of syphilis, shown in this figure. It's a very linear figure, and we'll talk about that in a little bit. But infection occurs by direct contact with an infectious lesion or an infectious site. The organism can go through intact mucosal surfaces or microscopic abrasions in skin, and it begins to multiply locally at that site. And after several weeks, averaging about 3 to 6, depending upon the number of treponemes that were introduced during the transmission, the primary stage appears. And this is characterized by a painless indurated ulcer called a chancre, which you can see right here, accompanied by regional lymphadenopathy. And this chancre will persist for several weeks and then will heal without treatment. And the patient may go directly into the secondary stage or may have a period of asymptomatic infection before the manifestations of the secondary stage appear. And that's usually seen as a rash, which is on the trunk and the extremities and the palms and the soles. This is accompanied by generalized lymphadenopathy. These lesions, each of which is a little focus in the skin of treponemes multiplying. This will heal without treatment as well, and the patient goes into the latent stage. And the latent stage, by definition, is asymptomatic, and but the patient remains infected. The vast majority of untreated patients will remain infected for the rest of their lives, and they'll die of something else. In the pre-antibiotic era, about 30% of people would leave latency and develop one or more of the tertiary manifestations, such as gummatous destruction of tissue, which you can see on the hand in that individual, cardiovascular syphilis, or the late, really serious manifestations of neurosyphilis, such as paresis, which can cause paralysis, insanity, and ultimately death, and tabes dorsalis, which is often associated with blindness and optic atrophy. Now, this is how I was taught syphilis, and it's probably how most of you were taught syphilis, but it's really a little different than that. It's called the great imitator for a reason, because it can appear in so many different ways. For example, the secondary stage, in addition to the rash, there can be constitutional symptoms, and there can be active involvement in infection of the liver, the kidney, and the stomach. And this can be subclinical or can be clinically apparent and the reason for presentation for medical care. About 8% of people with secondary or shortly after secondary syphilis will have ocular and otic symptoms. They'll have visual changes or hearing changes. And that 8% number comes from a paper that was published recently by Julie Dombrowski here at UW. The other thing to remember is that this is not truly linear. People who have secondary syphilis and go into the latent stage can have recurrent lesions, and this can happen several times, usually in the first few years of infection. But I think the most important thing that is not really appreciated, it's obvious that this is a disseminated infection, but I think the early dissemination of syphilis is rarely appreciated. And within hours to days of initial infection, before the primary stage appears, the organism invades the bloodstream, circulates, and disseminates to tissues throughout the body. And this dissemination is what sets the stage for the later stages, for the more serious manifestations such as congenital syphilis, which is infection of the fetus in utero, or central nervous system involvement. People in the first half of the 20th century and the 1800s as well really understood CNS involvement. But when benzathine penicillin became available in the middle of the 1950s and promised a cure of early syphilis with a single dose, people quit thinking about central nervous system involvement. And it was largely forgotten until 1988, when Ann Collier, Hunter Hansfield, and I published a paper in which we showed that we were able to isolate viable T. pallidum from 30% of patients with primary and secondary syphilis. And importantly, that we could isolate treponemes from the cerebrospinal fluid of patients who had already been treated with benzathine penicillin. And this was right during the big burst of the HIV epidemic. And Christina Mara came as a fellow during that time. She's a professor of neurology now, but she came to do an ID fellowship. And she and her group have explored, since that time, CNS syphilis and its interaction with HIV in depth. So if you look at this natural history, King always told me the best questions to look at are questions that come from what you see in the clinic, and I think he's right. And if you look at this natural history and you think of what the bug has to do to cause all of these different stages, what the host is trying to do to stop, to control the infection, you could come up with many, many questions. And I want to talk to you about just 4 of them today. The first is, what are the mechanisms of bacterial clearance in the primary lesion? In other words, how does a chancre heal without treatment? The second is, we know the organism persists. So how does it persist in the face of an effective immune response? How does the secondary rash appear? And can syphilis be prevented by immunization? But first I have to introduce you to my animal model. I work in a lab setting, and we use an animal model of syphilis, which is the New Zealand white rabbit, and it really faithfully recapitulates human infection. In terms of clinical manifestations, rabbits get primary and secondary syphilis. They have a chronic latent infection. The central nervous system is invaded during infection in rabbits, and we've seen rabbits with ocular syphilis. And the congenital syphilis model has also been developed in the rabbit. In terms of immune responses, the kinds of cells that infiltrate the lesions and the cytokines they produce are exactly the same in rabbits as they are in humans. And if you look at the repertoire of T. pallidum antigens that are recognized by human infection, it's exactly the same repertoire as is seen in the rabbit model. So we think this is a really good model. The rabbits are also outbred, which can be frustrating from an experimental point of view, but is appropriate because humans are also outbred, obviously. So let's go to the first question. What's the mechanism of bacterial clearance in primary lesions? If you look at the schematic on the left, this was derived from work that I did as a postdoc in an immunopathology lab at UC San Diego. The y-axis is relative numbers of whatever component I'm talking about, and the x-axis is time after infection. And if you look first at the solid line, that's Treponema pallidum. You can see the numbers of treponemes increase, and they reach their maximum concentration in tissue at the same time the primary lesion reaches its maximum size. Now, as those treponemes are increasing, there are cells that infiltrate the tissue as well. These are almost exclusively T lymphocytes, CD4s and CD8s, and those cells make gamma interferon, which attracts macrophages, which increase and activates them. And as the macrophage concentration reaches its peak, the treponemes disappear. So this is what we knew, and we have spent a lot of time since then working out the various aspects of what's going on during this. If you look at this graph, it's real data rather than a cartoon. These are days after infection, and the orange line is Treponema pallidum numbers done by quantitative PCR in lesions. You can see how rapid the decline is, and in blue is gamma interferon. So gamma interferon is really the predominant cytokine that we see in these lesions. In work that I did collaboratively with Wes Van Voris, we showed that these data in the rabbits were seen as well in humans, the same kind of cytokine profile in human infection. So clearance is an interferon gamma dominant or Th1 type immune response. George Starry, several years ago, showed that in humans there were Th17 cells as well. And Th17 cells can make gamma interferon. They also can make IL-17, and they're thought to be important for control of extracellular pathogens, which Treponema pallidum is. We didn't have the tools to look for Th17 in rabbits, but Th17 cells in rabbits, but we have looked for IL-17 just recently, and Charmy Gadornis, a research scientist in the lab, has shown that IL-17 is present in the lesions of our rabbits as well. So again, looking very much like human syphilis. And the clearance is actually opsonophagocytosis. So what is that? That's when a macrophage recognizes a bacterium that has antibodies stuck on the outside of it. That antibody makes the bacterium more likely to be ingested. It's like putting ketchup on a hot dog. It makes it more likely to be eaten. And so, and if you don't have that antibody, then the organisms are not ingested. So opsonic antibody is also an essential component here. And just to pull this all together, this is the way we envision local clearance of the organisms in early lesions. So T. pallidum are taken up by antigen-presenting cells, which process the antigens, present them to T cells. This is very simplified, but these T cells produce gamma interferon, which recruit macrophages to the site and activate those macrophages. And T. pallidum antigens are also recognized by B lymphocytes. With help from T cells, they differentiate into plasma cells, making antibodies. And the antibodies that can bind to surface antigens of Treponema pallidum will opsonize those treponemes, which will then be eaten and killed, ingested and killed by the activated macrophages. So the next question is that we know the chancre heals, but there are still treponemes left in the body. So how do they persist in the face of an immune response that has cleared really billions of organisms from that primary chancre? So this was a long story. This story took a long time to develop. At least 30 years ago, we knew that when we looked to see which treponemes were ingested in a phagocytosis assay, so we put macrophages in a dish, added treponemes that we had to extract from rabbit tissue because we couldn't grow T. pallidum, and a source of antibody. We knew that if we harvested treponemes from early in infection, when the numbers of organisms were still going up, they were very readily opsonized and ingested. But if we waited until the rabbit had started its natural process, which was clearing the organisms from the lesion, and took the treponemes that were left during that clearance process, they were refractory to opsonization and phagocytosis. And so we speculated that those treponemes that were persistent had somehow changed their surface so that they weren't recognized by the antibody. We showed that it had nothing to do with their activity on the macrophages, so it must have been that they had changed their antibodies and could be opsonized. We thought it might be antigenic variation, but at this point, 1992, we didn't know which antigens might be on the surface of T. pallidum, and we didn't know which ones might be likely to vary. So Arturo Centurion came as a postdoc to the lab at about this time, and it was about the same time that the genome sequence of T. pallidum was published. And Arturo got very interested in a 12-member family of proteins that he found in the genome, and called the TPR family. And very quickly, we focused in on one, TPRK, because it was the one that was most highly expressed during infection, and it was also the one to which the immune response first made antibody. So he began to look at TPRK sequences not only in our lab strain, but in other strains of Treponema pallidum. And what he found was that the sequences varied among strains, from one strain to another, and even within a strain, you could find multiple TPRK sequences. And so this is a cartoon showing a TPRK protein, for example, or even the gene. And what he found was that these sequence differences were not distributed randomly. They were found in 7 discrete regions, which we called V1 through V7 for variable region 7. We then did a computer prediction of the structure of TPRK, and you can see here that it has a typical beta barrel structure, which are what these bands represent, and that's typical of proteins that are embedded in the outer membrane of bacteria. And when we look to see where the V regions were localized, I hope you can see this with the light glaring on it, the V regions are shown in yellow, and they are all found on loops, which are predicted to be on the outside of the outside of the bacterium. And at that time, a graduate student in the lab, Cecilia Morgan, looked to identify where the antibody epitopes were. That is, what part of this protein was recognized by antibodies, and all the antibody epitopes localized to these outer membrane loops that were the variable regions. So this looks perfectly like an antigenic variation system. So you've got antigens on the surface, opsonic antibody needs to bind to them, the treponeme doesn't want to get killed, so this is my interpretation. So it changes that sequence, then the antibodies can't bind. So it looked very much like an immune evasion system, and we'll talk about that mechanism in just a minute. But Arturo was very interested in how the molecular mechanisms by which these variable regions changed. And so T. pallidum has a single circular chromosome. There's one expression site, so there's one gene locus for TPRK. And on the other side of the chromosome, there is a region, actually 2 regions, but very close together, where he could find small DNA sequences that he had seen in other strains' TPRK sequences. And what happens is that a piece of one of these donor sites, which we call these, is recombined into the expression site, creating a new mosaic or chimeric protein sequence. And this mechanism is called segmental gene conversion, and it's very much like the same mechanism that's used in the relapsing fever spirochetes. This is a spirochete as well, T. pallidum is. So the relapsing fever spirochetes, and in a protein called VLSE in the Lyme disease spirochete. It's also a similar mechanism that's used to vary the pillin gene in Neisseria gonorrhea. But another graduate student in the lab asked the question, does an antibody that reacts with this V region react with this now changed V region? And she showed that even a change of 1 to 2 amino acids in a V region could render that antibody unable to bind. So it's a very sensitive, it's very sensitive in terms of what the antibodies can and can't bind to. You don't have to change much to stop from being opsonized. So this is a model that we put together. If we were to infect a host, a rabbit, with a suspension of treponemes all expressing exactly the same TPRK sequence, so they're all blue, they're expressing a blue sequence, then the rabbit will make antibodies to the V regions of those blue spirochetes, so blue antibodies. But as the bacteria divide, they just naturally recombine to form TPRK variants. When the antibodies bind to the cells, the bacteria that are expressing their target antigen, those bacteria are cleared, leaving the variants behind, and so the antigenic variants are selected. This is typical immune selection. And a postdoc in the lab at that time, Lorenzo Giacconi, who's now an associate professor here, asked that question experimentally. He took 2 groups of rabbits and infected them with a suspension of treponemes expressing the same TPRK. And one group was the control, the other group was pharmacologically immunosuppressed. And this shows you time after infection compared to amount of variation in one of the V regions. V6 is the one that we looked at. And so you can see that in the control group, as time goes on and the immune response develops, you can see that the amount of variants, the number of variants also increase. Whereas in the immunosuppressed animals who were unable to make antibody, they continued to have very, very low variation. So immune selection really does occur in vivo, and we think this is one of the major mechanisms by which the organism persists in the host. It goes through this cycle of immune selection and making new variants again and again and again over the decades. The next question is, how does the secondary rash appear? We know that syphilis has sequential stages, and we wanted to know whether or not antigenic variation of TPRK had anything to do with the development of the subsequent stages. And so Tara Reid, who was an MSTP student in the lab at the time and who's now an ID fellow here, tested 2 hypotheses. The first is that each secondary lesion is seeded by a single treponeme, and that's exactly the same as a single bacterial cell on an agar plate. It'll divide again and again and again until it makes a colony. Well, a single treponeme in the skin will divide again and again and again until a secondary lesion appears. So that was the first hypothesis she wanted to test. The second is that secondary lesions are seeded primarily by TPRK variants. So the experiment that she did was take a group of 10 rabbits and infected them again with the suspension of treponemes, all expressing the same TPRK sequence. She let these chancres develop and heal, and as they healed, secondary lesions appeared. And she biopsied those secondary lesions as soon as she saw them and extracted DNA from them and sequenced TPRK from the treponemes that were in those secondary lesions. So the next slide, I'm going to have to explain to you, I think. These are DNA sequences. Each row is a DNA sequence, and the sequences come from 4 samples. First was the inoculum. This is the suspension that we infected those rabbits, that Tara infected those rabbits with. And then B, K, and N are secondary lesions. And the way you look at that, and there are 10 sequences obtained from each of these 4 samples. And so let's just look at this first one. The way you look at this DNA sequence alignment is that if the colors are solid all the way down with a dark colored background, that means that the sequences are identical. So if you look at the inoculum, V7 was identical in all the sequences she got from the inoculum. All the sequences from lesion B were identical. All the sequences from lesion K were identical. All the sequences from lesion N were identical. So this strongly supported the hypothesis that each secondary lesion comes from a single treponeme. The next question that she was asking was whether or not the secondary lesions were caused by variants. And so any place that you see a white background, like here or here or here, or here, or here, it shows that it's different from the original. So this clearly was a variant compared to that. This secondary lesion was a variant compared to that one. This one had an insertion and some other variations. So it was a variant compared to the original as well. And so this supported the hypothesis that secondary lesions are seeded primarily by TPRK variants. And here are the data. These are numbers of secondary lesions, and each rabbit is shown individually, showing you why outbred animals can be confusing and difficult. But 2 rabbits never got any secondary lesions. 5 of them got secondary lesions, and all of those lesions were made by variants. 2 rabbits had lesions, secondary lesions, in which the predominant most of the lesions were caused by variants. And then we had this one rabbit who got a lot more secondary lesions than anybody else. And in this particular rabbit, the inoculum sequence was found more frequently than the variant sequence. But if you look overall, of all the secondary lesions in this experiment, 65% were made by variants compared to 35% inoculum sequences. So we think that TPRK variation does play a role in facilitating the development of these secondary lesions. The last thing I want to talk to you about is, can syphilis be prevented by immunization? And the work that we've been doing over the years has led to this approach. We've identified antigens, we've looked at immune responses, we think we know what we need to develop a vaccine. And this is a collaborative effort with Caroline Cameron, who was a postdoc here working on syphilis with Wes Van Voris and me. She's now a professor at University of Victoria with Lorenzo Giacconi, whom I mentioned already, with 2 very talented research scientists, Barbara Malini and Charmy Gadornas, who've worked with me for over 25 years. And Mark Fernandez, who was a student in the lab and who's now a graduate student here. So our strategy is, we think we need to induce a gamma interferon dominant response so that we can clear treponemes. We think we need to induce the development of opsonic antibody. We think we want to prevent dissemination, because dissemination is what really leads to the most serious consequences of syphilis. And lastly, because of the antigenic variation that I've just been talking to you about, we need to use conserved regions of surface-exposed proteins so that we can overcome that variation and hopefully develop a vaccine that will be broadly active against many, many different strains of T. pallidum. So what antigens did we choose? We spent many years working with Wes and Lynn Barrett in his lab, identifying target antigens, expressing them as recombinants, immunizing rabbits, challenging them with infectious treponemes, and seeing whether there was any level of protection that occurred. The 2 that came to the top of the pile were both members of the TPR family. One is TPRK, and I'll talk about that in a second. The other is TPR subfamily 1. So the TPR family is divided into 3 subfamilies, and subfamily 1 has 4 proteins in it. And the amino portion, that is the first third of those proteins, are identical in all of these 4 proteins. They're also identical in other strains of Treponema pallidum. So it's a conserved region. And when Eileen Sun, who was a graduate student immunized rabbits with recombinant amino region of subfamily 1 and challenged them. You can see the unimmunized animals developed big lesions, which ultimately would go on to ulcerate, whereas the TPR immunized animals had just this faint erythema. These black marks are ink telling us where we had challenged the rabbit. And when you look to see how these lesions progressed further, 83% of the lesions in the unimmunized group went on to ulcerate, compared to 8% in the immunized group. And if a lesion doesn't ulcerate, it's not going to be as infectious as an open chancre would be. We got very similar results with TPRK fragment 1. Now you're going to say, TPRK, why are you using that? It's the variable protein. Well, the first, I don't know if you remember the schematic I showed you, but in the schematic, the first third of that protein is largely conserved. And V1, which is one of the V regions that's included in that particular piece, is the least variable of all the V regions. And so Cecilia Morgan had done these studies and showed that this was the most immunogenic and protective part of TPRK. So we're using these two antigens, both of which induce the development of opsonic antibody. Now, in terms of stopping dissemination, Caroline Cameron began working on a protein called TP0751 when she was here, and she's continued that work in depth since she's been at the University of Victoria, and has shown that this is a surface-localized lipoprotein, and it functions as an adhesin. It adheres to endothelial cells, it adheres to laminin and fibronectin. And it's critical in the dissemination of the organism, that is, in getting the treponeme into the bloodstream so it can disseminate, and then getting the treponeme out of the bloodstream so that it can cause disease in that new site. And we're lucky that the sequence is 100% conserved among all strains. And when she immunized with this protein, it induced the production of opsonic antibody, another requirement, and she showed that it inhibited the dissemination of T. pallidum. So our candidate vaccine has these three components. The TPR components are to protect against the chancre development and ulceration, progression of the lesions, which should reduce transmission. And 751 is to protect against dissemination, which should reduce the chance of central nervous system involvement or congenital syphilis. So how did we do? In our first experiment, we had 3 groups of 8 rabbits, unimmunized, and then 2 immunized groups, in which we were comparing 2 custom adjuvants that we worked with Derek Carter at API to develop, called Ribi Natural and Synthetic. And then we immunized the animals with there's a cocktail of these 3 antigens 5 times at 3-week intervals, and then challenged them intradermally with 10 to the 5th T. pallidum per site at 10 sites on their backs. And we looked to see what happened to the lesions. Now, interferon gamma dominant response is often seen clinically as a delayed type hypersensitivity response, just as like a tuberculin skin test, for example. And so we looked at the rabbits at 48 hours to see what proportion of the lesions had clinical DTH. And you can see that none of the unimmunized animals had delayed type hypersensitivity, but significantly larger numbers of lesions in both immunized groups showed clinical DTH. And because this is a surrogate for gamma interferon, we thought we should look for gamma interferon itself. So we looked for interferon message in the tissues. And again, you can see that the immunized groups are significantly higher than the unimmunized groups. So we got a gamma interferon response, which is one thing we wanted to look at. Now, the other goal of a vaccine is to limit the pathogen burden in the host. And so at day 19, we aspirated each of the challenge sites and looked by dark field microscopy at 100 fields per lesion. There were 80 lesions in this experiment. No, there were 240 lesions in this experiment. And so it took a long time to do this microscopy. And we looked at the numbers of treponemes that we counted. And you can see the number for the unimmunized group. And the immunized rabbits had significantly fewer treponemes visible by dark field microscopy. If you look at it the other way and look at the proportion of lesions that did not have any dark field identifiable T pallidum, so that we're dark field negative. Both groups, both immunized groups, had high levels. 61% and 32% of the lesions had no treponemes that we could see, compared to all of the rabbits here had treponemes, so that's why that's a 0. Because these are outbred animals, I thought it was important to look at each rabbit's data point. So you can see that the unimmunized group really had quite a range in terms of numbers of T pallidum that we could see. And both immunized groups had significantly lower, just as I showed you before. We also tested this by qPCR and showed a very similar pattern. So then we asked whether or not the lesions went on to progress, and we looked at what proportion of lesions had ulcerated by day 19, those are shown in the solid bars, and by day 30, which are shown in the hashed bars. And again, you can see that the Ribby natural group had significantly fewer lesions ulcerating compared to the control. The Ribby synthetic group was not significantly different from the control. When we looked at lesion volume, how big did the lesions get? Again, you can see that the unimmunized animals were significantly larger through day 25, and then all of the lesions began to heal by day 30, so there's not a significant difference there. The next question we asked was reduction in dissemination. And we did this, we know that the time between infection and the development of clinical manifestations is inversely proportional to the dose. So the more treponemes you put in, the faster they'll get a clinical manifestation. That makes sense. So we put together a standard curve of dose versus time to infection. And then when this experiment was over, we harvested the popliteal lymph nodes from these animals, minced them up in saline, and injected lymph nodes from one rabbit into one recipient, naive recipient rabbit. And then we watched those recipient rabbits to see how soon they developed clinical manifestations. And going back to our standard curve, we could estimate how many treponemes were in their lymph nodes. The unimmunized animals had 2,000 treponemes in them. The Ribby synthetic group had 1,000, so a 50% reduction in dissemination. But we were delighted to see that the Ribby natural group had only 200 treponemes in their lymph nodes, so that's a 90% reduction in dissemination. So this was the first time we did the experiment. I should point out as well that most of the hard work on these experiments were done by Barbara Malini and Charmy Gadornis, who are both research scientists in the lab. So the next thing we did was to try to reproduce it. A smaller experiment this time, looking only at Ribby natural and control rabbits, and we saw exactly the same patterns as I've already described to you. And we are now in the middle of another experiment in which will actually be, those animals will be challenged this weekend. And we're challenging with heterologous strains to see whether or not there's broad protection or whether it's limited to just our laboratory strain. So in conclusion, syphilis has a pretty complex natural history, and we have shown that bacterial clearance is by gamma interferon activated macrophages and opsonic antibodies, that persistent infection results at least in part from antigenic variation of TPRK, that TPRK variants are critical to the development of the subsequent stages of syphilis, and lastly, that immunization with this tri-antigen cocktail can attenuate chancre development and reduce dissemination. So I will just show you pictures of a couple of people. This is Caroline Cameron at University of Victoria. This is Arturo Centurion, who was the TPRK antigenic variation wizard. Lorenzo Giacchini, when he was a postdoc who did the immune selection experiments. Charmy, this was Halloween, and Barbara and Mark Fernandez, who were our research scientist and a graduate student, and Tara Reid, who did the secondary syphilis work. And this is Christina Mara, who's a close collaborator next door to me, and her group, who's worked on neurosyphilis. So with that, I'm happy to take any questions you might have.
Very nice tour de force, Sheila.
Thanks.
I'm interested in, because the molecules seem so highly folded and so on, how reproducible does the folding have to be on the recombinant proteins for your vaccine?
I didn't mention that because I didn't have time. We do refold our recombinant proteins, that because they're insoluble as they're made as recombinants. They have no structure, essentially. So then we do refold them, and we use a commercial refolding kit. We've looked at various ones, and we test every antigen by circular dichroism to be sure that we've got beta sheet there. So we're careful to do that. Yeah.
Yeah, Sheila, that's a great talk. Do you have any sense of the duration of immunity from the triantigen inoculation? That's
part of the experiment that we have going on now. We have been, all of the experiments that I showed you, we challenged 3 weeks after the final immunization. In this new experiment, we'll be challenging a group of rabbits 2 months after the final immunization, but we don't have time or money to let rabbits sit around for a year at this point. I think the vaccine's not quite ready for those kinds of studies yet. They're too expensive.
Do we have any insights into host immune variability, genetic variability, and sort of evasion? You know, you talked a lot about bug variability, but I was struck at how much variability there were in your rabbits. And in humans, epidemiologically, there's a lot of difference in races and that sort of thing. And I don't know if that's thought to be environment or if there's some sort of immune
Yeah, host variability is there. I mean, some people get tertiary syphilis if they go untreated. Other people control their treponemes just fine. We don't really have any idea exactly what components are important. Christina has done some work on with neurosyphilis, with your HLA. Was it HLA? No, it was a. Sorry? Yeah. You want to talk about that?
We looked at some with, sorry.
Here.
I could just talk really loud like that.
There you go.
We looked with Tom Hahn, at some TLR polymorphisms. There's a common TLR2 SNP that it decreases the likelihood of getting neurosyphilis, the ANMISH TLR1 SNP, and a TLR6 SNP. We did whole exome sequencing, looking at the same question, and it came up with, I'm trying to remember what we came up with. I can't remember, but we came up with another SNP that may actually be protective as well. ICAM1.
King.
So while you and your team are making such impressive progress in figuring out how to generate antibodies and control and prevent syphilis, the syphilis is working pretty hard against you, and it's pretty smart too. And as you know, and maybe not everyone here knows, we now have a new epidemic of syphilis that is spreading around the world in what we call the PrEP era, with pre-exposure prophylaxis being increasingly made available. There is more sexual risk-taking and increasing rates, not just of syphilis, but lots of the STDs, including gonorrhea and so on. So Sheila, how are you going to cope with that?
I'm not going to cope with that. That's Matt's job. He's the public health guy.
Well, I'm just wondering if there is more rapid transmission occurring amongst larger numbers of people. How does that, what does that call for in terms of increasing attention to diversity in the population and so on? I would expect we're going to see more diverse populations as we have more people getting infected. They tend to be coming from the same groups that were acquiring HIV, but I think there is a lot of diversity in terms of those becoming infected too. So, does that make any sense?
Yeah, but are you talking about diversity in the humans or diversity in the bugs? I think
both.
Well, the bacteria, if from all the genome sequencing that's been done so far of T. pallidum, there's very little diversity. I mean, you can make a tree, but they're like 99.9% identical at the genome level. So there's not a huge amount of diversity from one strain to another, except in certain hotspots like TPRK, for example. That's the only place that diversity is being seen. But how that translates to the rapid spread. I don't know if syphilis is really being spread more rapidly or if people are just having more partners. I think it's more behavioral than at the bug level. Yes.
Sheila, what do we know about the difference in the human immune response compared to rabbits, and what would need to happen, broadly speaking, to start looking at an immunization in humans?
Well, the mechanisms of immunity are exactly the same. The cytokines that are made, the need for antibodies to get phagocytosis of T. pallidum with human macrophages and requires human antibodies. So the immune responses are exactly the same as in the rabbit. So I think that part's translatable. What's not translatable is the adjuvant that we use. We're using an adjuvant. We tried in our lab 14 different commercial and custom adjuvants. Some of them gave no protection at all with exactly the same proteins. So the adjuvant that we're using is one that has a TLR2 and a TLR4 agonist. And the TLR2 agonist is really critical to that, to protection. And the components, you know, at the beginning I said that we were looking at a synthetic version of the adjuvant and a natural version of the antigen. Well, the synthetic version was an attempt to find one that would be more adaptable to be used in humans. And it was not as protective in the rabbit. But I think what our goal is to find the antigens that are important. And then you can go to humans with different adjuvants that can be used in humans, because rabbits react to adjuvants differently than humans do. So I think we're trying to identify the antigens at this point, and then the next step would be adjuvants for humans. But at least we know what kinds of immune response we want to generate, and that will help guide you in the development of the adjuvants. Yeah.
This question for you, and maybe Matt can chime in. How much transmission occurs after the chancre has healed in humans?
Probably not much. What we know, though, is that people with early syphilis have treponemes that can be identified from rectal swabs, from oral mucosal swabs, and DNA can be found in urine as well. So that's all done by PCR. So does DNA equal infectious organisms? We don't know that yet.
I'm just curious about all the energy, evolutionary work that the treponeme is doing without much return in passing on its own DNA.
Well, it has the primary stage, it has a secondary stage with those lesions. It can do that again and again. And if this mucosal shedding that we're beginning to see data for is important, then it's got months and months and months to transmit itself, which is longer than many pathogens have. So, yes.
So, are there any receptors known that are required for host entry? And, in terms of TLR adjuvants are thought to be useful, but are there PAMPs that are recognized from T. pallidum?
Well, T. pallidum has a lot of lipoproteins, and those lipid portions are what's recognized by TLR2. At least several, the ones that have been looked at have been recognized by TLR2. I'm not quite sure what's being recognized by TLR4. There's no lipopolysaccharide in T. pallidum, but which is a typical TLR4 agonist, but we don't know what's triggering that. Judy.
Sheila, that was a great talk. Thank you. You didn't talk much about treatment, and one of the that would be in the not much category. One of the nice things about T. pallidum is that we don't see a lot of antimicrobial resistance emerging so far, but there's always been this challenge of treatment, particularly in privileged sites. You talked about dissemination of the infection. Would you like to say anything about that aspect of dealing with syphilis?
Yeah, I'll say a couple of things about treatment. Penicillin, as I said, is still the drug of choice. There's been no confirmed penicillin-resistant strains of T. pallidum yet. But over the last 15 years or so, if I'm remembering my years correctly, there has been a very rapid emergence of macrolide-resistant T. pallidum, and essentially all the strains, the last time we looked, nearly all the strains in Seattle were macrolide-resistant, and this is true around the world. So azithromycin, which was thought to be maybe a single-dose oral drug that could be used instead of 2 shots, is no longer relevant. So, the rest of your question was, sorry?
Treatment in privileged sites?
Oh, in privileged sites. Yeah. The privileged site, this organism goes everywhere. It goes into the fluid in the eye, it goes into the fluid in the ear, crosses the placenta, the fetus being a privileged site for many antibiotics. It goes into the central nervous system, the blood-brain barrier, stopping a lot of antibiotics from getting through. I think it's a really important issue, and when we showed that we could isolate T. pallidum from the spinal fluid of people who'd been treated with benzathine penicillin, we know that benzathine penicillin does not cross the blood-brain barrier. It doesn't touch organisms in the central nervous system. And so is that important? And this has been a raging argument in the field for a long time about how frequently you need to do lumbar puncture to look for CNS invasion and what early CNS invasion means. And I'm not the best person to talk about that. That would be Christina. But I think we really do need to be cognizant of the fact that people can still have treponemes in their central nervous system after they've been treated. And for most people, I mean, you don't see a lot of people with past syphilis walking around with tertiary syphilis now. They were treated with benzathine penicillin, but even if you don't treat anyone, if you look in the pre-antibiotic era, most people were able to either clear or control their CNS treponemes through their immune systems. That's why when HIV and syphilis came together chronologically, there was huge concern, and Christina's done a lot of work on that.
Okay, let's give Sheila and Lukehart another round of applause.
Thank